So this question is not quite on point, but I don't know how to ask it better. Do you know the answer to my confusing question?
I think so. Maybe it's just the binding sites that we have.
Binding sites. So if you think about a protein being, I said that the interior of a cell is like a solid state. I said it's like a clock. But still, this protein is surrounded by water, by liquid, which essentially is water, right? So it is dissolved. It sort of swims in a tiny shell of water. Again, there's another molecule next to it, but it is dissolved, so there's a sort of monolayer of water, a little bit more than that. So there really is water, or whatever is in that cell different. So which part of the protein sees it? The binding site, yes, binding site. What are you binding to what? If you're swimming around in water, where do you feel the water? It's a very dumb question. No.
Membrane of the cell.
The membrane of the cells, but the membrane of the cell, in fact, is lipid. And what we are seeing here is not a cell swimming in water. What we're seeing is a protein swimming in water. So in some sense, what's the analogous thing to the membrane of a cell in a protein that swims around?
Surface of the protein.
Just the outside, right? So that's what we then would possibly expect, that in fact what correlates is the surface, not really the overall amino acid composition. So let's just look at that. What I show here is the residue composition on 2 and 3 principal components. The principal components are such, and I'm sorry for the quality of the slides. Maybe for these few I switch off the light. They are old slides here from '98. So this one here in light green, those are the nuclear proteins. In red is cytosolic, and blue is extracellular. So you do see, and again the principal component analysis here is such that each eigenvector is a sort of composition of all the vector of all the compositions. I cannot even read anymore what it says. That optimally separates these points. Now you can see exactly what we said, that the subcellular localization is different between according to amino acid composition. But let's get now into the core residues. The core residues is the inside of the protein. And the inside of the protein, again, despite the low quality of the slide, you may somehow see that this separation is less clear than that separation. So yes, there is still sort of a green part here. There are some red ones, but now red and blue is pretty much close to each other, and there are actually green ones in here. Then there are suddenly green ones up here, blue. So there's much more of a mixture here. Now what I should look at is sort of the complement. The complement is essentially the surface residues, and that is here. Now you see the extracellular, blue, nuclear, green, and cytosolic. Now the first thing I believe that you very clearly see here is that this separation is much better than this separation, certainly better than that one. And that one is better than this. So meaning if you take all the residues, you do better than if you only take the core. And if you take only the surface, you do much better than taking all. So the signal clearly is in the surface, right? That you immediately see by these 3 PCAs. Then there are some interesting points here where you see a green, I'm telling you that you could see it if my figure would be better. There's a green in what you may call the red cluster here. There's a blue here in the red cluster. So there are intruders into some other cluster. When we looked at these in particular, what we saw is there were sugars put upon those. So essentially what happened here is that by putting a sugar, you change the biochemistry. So you change the surface. And by putting the sugar, you can actually make the protein be adaptable to 2 different environments. You put the sugar in, and it lives in a different, so the extracellular protein here sort of lives in the cytoplasmic world with the sugar. And that's exactly what happened. So we suddenly see glycolation, putting a sugar on it, as a simple mechanism by which you can change the surface behavior and actually adapt to a different environment by just putting a sugar. So again, proteins do put sugar on to be well in different environments. One way of showing that signal here is by plotting the overall surface composition, and you see the height of the letter is proportional to how much that is used. So this is the actual ratio, not the ratio, this is the actual percentage. And you do see immediately what is so nice about these letter plots. You immediately see that the three differ. You don't need much on it. Okay, so clearly there is a signal in the surface composition, and that signal is in fact mostly in the surface composition. So there's a signal in the amino acid composition, but mostly in the surface composition. Knowing that, in principle, we should be able to set up a prediction method, because what I showed so far is a PCA. A PCA, if you want, is the simplest way of predicting, right? The simplest way, you simply cluster your points according to the principal component analysis, and then you have a very simplified prediction method. I'm not even showing you the performance of that. And the work here is from Rajesh Nair. So many years later, so this work was published in 98, and Rajesh in 2002 redid that on a larger data set. On the larger data set 2 years later, suddenly the signal no longer looked as well. So this is surface, and this is everything. Okay, everything is clustering less well than this, but the clustering no longer is as good. So what happened is, in those 4 years, many proteins had been added to the database that were much more confusing in terms of the signal. So what we had published as a signal, I believe, is still true, but a more recent database had a lot of, let's call it, pollution in there. So the simple SVM no longer is enough. By the way, there is the surface composition, amino acid composition, and here is if you look at secondary structure only. So we asked simply, instead of saying what fraction is alanine, we ask what fraction is helix. And we wonder whether that maybe relates somehow to the degree to which we can predict subcellular localization. Let me begin with an insert and have a brief run through neural networks. So neural networks essentially are devices that do a very simple computation. You begin with an input, here in blue, you have an output. So in this particular case you have two units, the first one here and the second one. They're connected to the output with a connection here, J11. Now if you come in with an input that is essentially a vector, another vector that you have is spanned by these two connections here. The connections are the free variables in the game. So you multiply two vectors. This is geometrically what you do. Multiplying two vectors, essentially you look at the projection of one upon the other. And then you apply trigger function. So let me, sorry, that's the geometric interpretation. The arithmetic interpretation is you multiply, you take the output of this blue here, what comes out of this one, the value of the input unit, multiply it with the connection, take this one, multiply it with that connection, this one sums these two products, okay? And that essentially is this geometric operation. And then what comes out of the green one here is an application of a trigger function, where you essentially ask, is this projection lower or higher than something? You put some sigmoid function that when it's very highly positive, it's 1. When it's very highly negative, it's 0, for instance. Or you could put any, so this happens to be hyperbolical tangents. You could even fit this with linear functions. But essentially what you have to create here is something that sort of switches into two different states. And that ultimately, it's called neural network, because the idea was that neurons aggregate signals, electrical signals, and when the potential of the neuron is above some threshold, they fire. Okay, that fire is exactly this corresponding to reaching the 1. That's the idea. Now, what you can do is essentially the next point, the way you train a neural network is you compute some error, and the error is you train it with data that you know, so data that you want, so for which you know the mapping from input to output. You compute what the network has output minus what you want and you square it. Why do you square it?
So you don't have any sign stuff.
Yes, so you want something, the error, whether the wanted output is 0 and so the direction of the mistake is not important, the magnitude of the mistake is important, the absolute value of the mistake counts. You could have done here an absolute value. This is easier to differentiate. That's the original reason why people use the square initially. So then what you in fact do is you compute this error and you try to change, if that were my first step here, you try to change these connections such that this error decreases. Step by step. You present examples at different time steps and you try essentially in an error landscape. So this is this axis here shows the connections, this the error, and the idea is there are some connections for which the error is lower. And training essentially means adjust the connection such that the whole system does less of an error. Okay? And so you, through various rounds of training, I'm not going to go through the details here, you simply change the connections. And what you can do is you can distinguish by introducing a line these two types of points. So the dark and the open. And all of these points here are equally correct. None of them makes a mistake. And in fact, depending on what the initial conditions are or what some details are, you find in fact different lines. You're not always finding exactly the correct line, but in this particular case here, the assumption is you find a very correct line. So assume we had these 4 points to distinguish. Can anybody show me a line to distinguish those 4 points? Yes?
You have to use hyperbolic functions there.
Yes. So those are 2 different answers with two different ways of doing it. This is one way of doing it, and hyperbolic function is another way of doing it. This is in fact one way of doing it. Yes, another way of doing it, if you don't use hyperbolic function, is you essentially introduce another line. So you could do it with 2 lines, straight lines, and then say everything that is within these 2 lines. In the language of the neural network, how do I introduce a second line? So to speak, and this is really a very, very intuitive understanding of it, so to speak, you introduce a new layer. In red here, called hidden layer, simply because the output has a reality, the input has a reality, and everything else is computation. Okay? Now going back to these deep learning methods, essentially now a 3rd line, a 4th line, a higher dimension is not added by additional layers, but by blowing up the number of hidden units. Okay? So essentially the way you can construct these simple networks is you just have a very, very large number of hidden units, and you can sort of compute any, or you can essentially solve any solvable problem with it. The reason why now in deep learning people go into several layers is simply because layers have a meaning. Typically those different layers refer to, historically they come out of the imaging processing idea, so those refer to particular image filters, if you want. So they're particular interpretation for what each layer does. And that's the reason ultimately why deep learning operates with many layers. In principle, from the computation, you could just do it by having one layer and blow it up. But again, what you achieve is that you do some meaningful processing by introducing particular layers. The computation is the same as before. I said you multiply the input with a connection here, you sum over all that come in this particular case. This one sums these 2 here, and this one sums from this and that, also 2. So then you apply the trigger function, and that is essentially the output that comes from here. You multiply again with this connection here. At this point you sum up the 2, that's this one, and you apply another trigger function. As simple as that. It's the same as before, it's just a repetition of the same equation, right? Now, the way, again, this is the output, you do the error in the way we showed you before, and then training means you go downhill. Now, downhill, as you see, you might encounter minima, and so you go over those minima essentially by allowing an additional sort of uphill move. So the equation that I show here is simply a gradient descent. So you say that the change of the connection at a particular time point t plus 1 is proportional to the gradient of this function here. So essentially it means you go downhill, okay? This is what this one here says. That's the gradient. The epsilon here means you do a step, a little bit of a step downhill. You're not jumping the entire way here, you do a little bit. Okay, that's what the epsilon controls. And then there's this plus alpha thing, that is the uphill move. Essentially it takes it from the change before and thereby allows you to go uphill. Okay? To not be trapped in minima. Now these two free parameters here are such that typically, so this is a cooking recipe thing, you typically take that to be 10 times smaller than this one here. And the epsilon you make as small as you can. Typically what you do is when you train the neural network for the first time and try to see how it works or what works for your problem, you have a very big epsilon, big means close to 1 or something like that, and you try to really jump down as fast as you can just to see, can I train this? How many hidden units do I need? Is that problem at all doable? Do I in fact have the right input units? And once you know that somehow, yes, you can use 20 amino acid composition and you use, say 10 hidden layers and somehow you can do it, then you sort of train slowly, and by training slowly you avoid that you're sort of trapped in minima. That's the entire idea. We have discussed this briefly here. If I plot on the x-axis training time, on the y-axis I plot the performance or the accuracy to the degree to which I learn the problem, then what I see, can somebody explain what I show? Oh no, wait a minute. Yes, I have two curves, and I have something that says overtraining. What is the green curve? Yeah? Yes. So what it means essentially, for the training data, I do better and better and better and better and better. Which means that in fact my input units are, or the way my input features suffice to solve the problem for the data that I put. It can learn it. So in this particular case, close to 100%. Okay? The blue one here is sort of the leave-out set. You may call it test set, validation set. There are different names for it. And in this one, there is a maximum point. After that, it falls down. And the interpretation of that is that we overtrain. Overtrain means essentially after this point you only learn, so to speak, by heart for the training set. You're no longer generalizing. Now typically when we see an image such as this one, we assume that before this point we were in the phase where we generalized. After that we are not. It's not quite right. So at some point most likely we began losing generalization earlier. That's the first issue. And the second issue, in reality, I believe we have never seen anything like this. Okay? This is typically what you see. I'm not sure whether I'm going to show you some curves, but typically what you see is something where over a very long course of the training time, you numerically can see that one of these blue values is highest, but very often you have curves that look rather, let's say, flat. So, I don't have a blue, yes I do have a blue. So the blue line would be over the training time typically something like so that you're not really sure what is the point at which you lose it. So yes, here's the first point where you have some sort of local optimum in the training set, but maybe this one is higher. So at what point do you really begin to generalize? At what do you really stop to generalize? At what point do you overtrain? In a curve such as this one, although in the way I drew that here, this one is the highest point. But you have passed through a bunch of maxima here, so possibly every single time was the end of some generalization of possibilities. So in reality the story is a little bit more tricky. And in fact tricky means we actually don't know. Okay? It's tricky in this particular case does not mean that when we think about it well enough we have a solution. There is no solution. Okay? It's not at all clear to say, well, this one here loses its generalization. People tend to stop at the highest point, meaning they tend, in my view, to overtrain. because the highest point here most likely went through a couple of non-generalizer part anymore. Anyway, and this device you can, for instance, use to predict secondary structure. When you do secondary structure, you essentially code the 20 amino acids. You take a sort of fragment of a protein. Well, I'll show you that later at some point. And this is one of those examples here. It's not as bad as the one that I showed. That is the one that used to be blue here. That's the validation or test set, leave out set. You do see that the training works fairly well. You do have an optimum here fairly early on, and this is not surpassed. So this one, if that would be higher, would be more similar to that. But we have seen things like that one too. Okay. What's different in the support vector machine? So let's begin with the math. The way we think about the whole story is exactly the same. So if you want, the idea is that you sort of project your problem into a hyperspace, and you try to find hyperplanes to separate your points. That's true for the neural network, that's true for the SVM. Does anybody here in the room know what the difference is for the SVM? So let's just show you the first sign, and it goes back to Vapnik. So it transforms the original problem into a transferred problem, a higher dimensional problem. So that is essentially the same thing. You were about to say something?
Was like the first thing that is a difference is the main goal between the neural network and the SVM.
Is the mean?
The final goal
is what?
Because like the SVM cannot do regression. But
well, yes, they can. They can, again, if you have enough data, you can learn any problem, right? You can learn regression.
but you mostly learn a separation.
You learn a separation. Mostly learn a separation, yes. But again, so given, if you were to teach it a data set that is sort of taken from regression, you can learn it. So it finds a different point, you're totally right. But nor does the neural network. So neural network and SVM in that sense are very similar. Yeah? Anybody? Yeah?
So maybe this is a constraint optimization, because we want to separate the classes, but if we want also to have a maximum margin. While with the neural network, we just try to optimize, but we don't have any constraint with that.
That is true, and in fact, this is exactly this point here. So you're trying to have a margin, a minimum margin. You're trying to set points apart. That is true. However, mind you, the more the error goes down, both for a validation set and the training set, the more, in fact, in a neural network, you will take these two types of points apart. So the margin between the two types of points will increase. Right? In some sense, it is true in the language, in the image of the way Vapnik introduces the reason for building SVMs is always focused on. The support vector machines are exactly the ones that sort of increase the margin. In principle, that's what the neural network does. The crucial bit is a different one here. The crucial bit really is that the argument is some points are trivial. These three here, or these three. Any initial training step would sort of classify points that are so far apart. Vapnik says what we should focus on are the more tricky ones. So find the red that are closest to the green and the green that are closest to the red, and try to put the margin, your support vectors, exactly there. Those most difficult to classify, those should define the interface, the margin between the two. Exactly where these two clouds of points are most similar, that's where you have to work on separating them right. And that, in fact, is sort of the underlying philosophy, and that philosophy has two important implications. One is you essentially need fewer points, because most of the points you're not even using for training. And indeed, SVM can get away with 10 times smaller data sets. Okay, that's the absolute great aspect of SVMs. The downside, can you see the downside? Oh, yeah?
The noise.
The noise, exactly, Sylvia. So when you have a mistake, imagine, so we're picking the points, the red ones that are closest to the green. Well, if you had a red one that is wrong, right? Odds are very high that that will be very close to the green, just because it's wrong, right? If in that projection you have clouds, a mistake falls into the wrong cloud. And you would pick exactly those points. So there's some way in the SVM that you can say, well, I'm going to exclude some points. True, so there's essentially an error margin. But ultimately, that is a problem that the SVM, so the more noisy it is, the more problems an SVM will have. And in fact, will not be able to do it, full stop. Okay. So let me just sort of have the idea of machine learning through slides from Teresa Word. And so her idea is, so she's sort of, for her the alarm clock is important because of some reason or other. And she tries to machine learn somehow what's the best way of waking up in the morning. So one way of a wake up point essentially is by just scanning how many hours have you been awake yesterday. So the more you were not sleeping yesterday, most likely the more sleep you will need overnight, right? And then you sort of can do some experimentation and you see something like that. And you see, well, is that a good feature? So at least there's some message that says maybe, sort of. Can somebody else say something? Let's turn it around. Is it a bad feature? What is the definition of a bad feature? Can somebody give me that? So again, obviously I want to classify green and red.
A bad feature is not distinguished.
It's not at all distinguishing. Essentially, it's completely random. That means if you had something like red, green, green, red, red, green, green, red, or something like that. So you had an alternation, complete alternation. And I explicitly did not say green, red, green, red, green, red, because then at least there's another sort of possible feature that you can restrict. But no, so if you have a complete mixture, this is not what you have. So you could put a threshold here and will be mostly right. You put a threshold here, will be mostly right. So we clearly have a feature that is not in a sense 100% right. But it gives you some signal. Okay, now for machine learning, I would argue this is a very good feature. Why do you believe I would argue that if we had a feature such as this one here, why do you believe I say for machine learning this is actually not so interesting?
Because it's way too simple.
Yes, we don't need machine learning for that. So all we need is a very simple threshold. No machine learning needed, okay? In that sense, so this kind of feature is in fact exactly the sort of thing we are looking for when we set up machine learning devices. Now, another question or another way of looking at it could be hours of sleep until getting up. So how many hours did you sleep at night? This is another thing that sort of may be relevant for how well you feel when the alarm clock tells you now it's time. And again, you see the idea in this particular case. With these two, you find a relatively simple separation. Okay. Now, did we talk about the binary classification story in the on the whiteboard, in the chalk talk? So essentially the SVM, another important, let's call it constraint, out of the box the SVM is essentially a device that classifies binary problems. While the neural network can in fact easily have 3 different classes. That's one issue. Now let's get back into this issue of subcellular localization. We did discuss it. How can I take a binary device or how can I sort of machine learn three classes, cytoplasmic, nuclear, extracellular, with a machine learning device? Well, one answer is you could have a neural network that has these three output units, extracellular, nuclear, cytoplasmic, or you could essentially put it into a binary network, or three particular networks where you have in the first one two output units, extracellular against everything, nuclear against everything, and cytoplasmic against everything. And in fact, now one way of finding the final prediction is essentially take whatever has the highest probability of these three. So you have three different predictions for every single protein, and you simply take the one where signal, so the positive unit, is highest with respect to the other state. Okay? Is that clear to everybody? Good. What else could you do? I mean, again, you could also take three output units, but I assume I would have drawn an SVM here. So a device that is more intrinsically binary. So assume I really want to make do with sub-solutions that have two output units or a binary classification. By the way, there's another point here. I do show two output units. Do I need two output units in a neural network? Can somebody explain to me how I can get away with one? Francesca, Tobias, today you haven't spoken, yes?
You can just code it as 0 and 1, for example.
So essentially then you would say, if there is one output unit, you would simply say whatever is closer to 1 is meaning cytoplasmic. Whenever the output unit, the single output unit is closer to 0, I call it non-cytoplasmic. So the reason for using 2 typically is anybody can guess some reason? Why could 2 output units be better than 1? Yeah?
So I guess for 1 it's more comfortable to use, because you can just compare the output vectors and you don't have to recode them.
I'm not entirely sure what you mean by recode, and I'm not sure.
For example, if you encode them as 0 to 1,
yeah,
you need to put a threshold, and if you want to compare the three individual probabilities, you need to extract the relevant probability.
But you use 2.
No, if you use only 1.
Why? When you have only 1, 0 is between 0 and 1. And whatever is a different signal is, in fact, it's the other way around. You're totally right, it's just the other way around.
Oh, yeah, good idea, I'm not sure.
So you're just arguing for the opposite of what I said.
Yeah.
So my question is, why would you want 2? So this would be a reason why you would want 1. Because for 2 you have to sort of do a conversion, essentially it's a normalization, where you sort of divide, you take this, divide by the sum, you normalize the value, and then you essentially transform it to a probability that is on the same scale between the 3. Because the way they train, if you have 2 output units, they do not necessarily have to sum to 1. You have to ascertain that through normalization. But that's a relatively simple process. You're right. That could be because you're too lazy to sum up 6 values and divide by. Okay. But what is the reason for wanting 2 output units rather than 1? But again, thanks, because it is important that you have to normalize them. So I'm making a joke of it, but you have to remember that, because otherwise you could actually not compare them to each other. And I usually don't say that. Why would you want 2? Why could it be an advantage to have 2? Yeah?
You could plot the results with 2.
So you're anyway, I tell you argue. Okay, yeah. Sorry. Good. You could plot the results in a scatter plot, for example. So plotting is one way of looking at it. Another way is, we will get back into that. There's something that I typically refer to as the reliability, which is sort of the strength of the difference, and it is more meaningful if you have 2. You can plot it in a scatter plot. You can plot it in a 2D plot. Yes, that's one way of saying it. Something else. Well, it could be that for a particular protein you really get the signal in both directions. And that could be very meaningful because it is a protein that in fact typically has a sugar. In this particular case, without the sugar, it looks like it can or cannot. So it's a very uncertain classification. And this device could in principle learn to have strong, both units close to 1. I'm not saying that is the standard case, but by in fact having a bunch of additional units here, or free parameters, I could learn that some input patterns map to 2 output units that are both on. Most of the time, one is on, one is off, but by 2, in 1 unit you could never see that, just by definition, right? By 2 you could, and you could, so in 1 unit, essentially what you then would see is a value of 0.5, right, undecided. But here you could distinguish between undecided that comes from both units being close to 1, and undecided that comes from both units being close to 0. So that carries additional information in principle, okay? Yeah?
I don't understand, because at the end, in the last 2 neurons,
Yeah.
don't we do probability, so we do like softmax?
Yes.
So how could they both be?
No, but wait a minute. So this layer is still completely learned. Oh, the softmax, by soft you mean the final function?
Yes, so
but the final function is a sigmoid.
Oh, of both, because usually when we have more than 1 class, we if we have 2 neurons, we use the softmax.
Maybe not.
It's okay.
Because there is information. There is important information contained in this.
I see that.
Good. Okay, but my question continues to be, so be it as it may, you could somehow find the one with the highest value here. That's one way of deciding which one is the winner, which one is the final prediction. What else could you do? So again, the problem is I have 3 different networks. Each one has a prediction, and I want to find out what my final prediction is. So for each of these 3, I know it is cytoplasmic or other, it is nuclear or other, it is cellular or other. But now I want for the one protein to have one final assignment. And again, the simplest way is to choose the highest. What else could I do? Yeah?
You could use machine learning on those outputs as well.
Is that what you wanted to say, Sophie? So essentially one way of doing that, I could have a second level here, where in fact the output from the first one, so the 3 different networks come in. That's one way of doing it. Okay. Here it's shown in a slightly different way, where we also possibly can use different types of information, but we could do the problem that I had before. Yes, that's one way of doing it. Now, another way of doing it, in fact I should have done that, this is the story that I had. Somehow another way of doing it, but it's related. I could sort of carve this whole thing into a hierarchy. And in this hierarchy, what I mean is I could, rather than sort of seeing all of these 4 different, now I have 4 states here, I added in this slide the mitochondria. So these are the 3 that I had before. Rather than just taking the highest or putting a machine learning device on top, which one is best, in fact I could map them to a hierarchy of the following type. So again, what I briefly mentioned is that from biology we know there is an exchange between nucleus and cytosol that is gated, special somehow. Then there is a major sort of what people call a path, the secretory pathway, where proteins have a single peptide when they get here. And then there's a particular exchange with the mitochondria. Now what I could simply do is put a hierarchy that reflects this path. So the way that could, for instance, work is, first we asked, is we train here, this particular case, what I show is an SVM. We train an SVM that simply says, or asks, is my protein going to be on the secretory pathway? Okay? So here we could, for instance, use a single peptide prediction. Or we could also use something else. But this SVM looks at all proteins and simply says, am I going to go into the middle path here? Am I going to go into this pathway here? Or not? If I go into the middle pathway, then there are different alternatives. If I go on the other one, I could go to the nucleus, or I could go on cytosol or mitochondria. And each of those here is a separate machine learning device. In principle, it's similar to the one before, except for now, we no longer have the decision what is the highest value. We don't have a machine learning that learns from 3 or 4 initial outputs, because at every single point, I have only one decision. Okay? Now, I said that one way we can sort of construct this one here is by following the biology. And in fact, the beauty of following the biology, if somebody comes one day with new data to you and says, well, I have data about chloroplast, then all you need to do is add another component at the end of your hierarchy. You don't have to redo anything that you did before. Right? That's the beauty of it. Can you see what is a disadvantage of this solution? Yeah?
In the previous way of solving the problem where we had 3 neural networks, in case that 2 didn't agree, we could at least see it. Here, if we have, for example, in the uppermost level, an error, then the whole prediction is going to be wrong.
So there are 2 issues here. And I begin with the last one you mentioned. If you make a mistake on the highest level, you can never recover from that. The second thing is, you're not seeing whether 2 are similarly high. At every single point, that part of information what you lose is because at every single point you have only binary decision. Either this or that. You're not seeing that it could also have been something else. That you lose, okay? Now, I said that we motivate this particular choice here, the first is secretory pathway or not, by the biology. Sophie says, what is really bad if you make a mistake up here, because you cannot recover. What does that mean to you, how you choose what is on the top level?
The most general specific feature that actually distinguishes the...
You are totally on the right path, but I want you, Reza, to say it more clearly. You said the most specific, and then you said the most general. This is a contradiction in Suhu, right?
I mean, not specific. I would say that probably some of the features that are actually totally different and...
We're not talking about features. We're talking about distinctions. Feature typically is input feature, right? What we're talking about here is particular binary solutions. So we have, in this particular case, on the whiteboard, what I show you, in fact, is no longer 2 classes, 3 classes. I have cytoplasm, I have the mitochondria and nucleus, 1, 2, 3, 4, 5. At least 5 different states, okay? So, and I have in fact 4 distinction devices. How do I choose which one comes first? And I still leave it with you because I believe you have the answer. I just want you to make it more explicit. Or do you not? Maybe I confused. I had the impression when you first said it, Reza, that you had it. Do you? No. Okay. Francesco, you wanted to?
So I will put something that splits.
Or you would do something else?
Like in the way of a tree. Like it
Well, that's in the way of a tree. That is a tree.
Yeah, like where I have where my division of the successive data is the biggest. I don't know how to say it actually.
But that's the crucial bit. You have to find a way to say it. I believe Reza had it also right, but he didn't know how to say it. You're both close. Sylvia, you...
I think I'm thinking the same as him. Like the
Fair enough. So say it right.
You can take the 2 classes that has a better margin, a more clear margin.
Margin is the wrong word. You're all touching on the point, but you're not putting your finger on the point. Yes, you're right. But what does it mean? There's typically another way of calling the margin. What does it mean if the margin is higher? That's true.
They are different.
Yes, they are different. They're all different. Otherwise, you couldn't classify. Yes.
We already use quality metrics on these SVMs for training, so we just take the one where this quality metric is maximized, probably. Like
The quality metric. What is the quality? Exactly. Sorry. I take it you had the same on your mind. So yes, you were all right, but the point really is performance, right? The performance is best. The margin is highest if the performance is best. But they all have to have a margin if they are better than random. So the real thing is you put on top the one that does the fewest errors. Because if in the first step you cannot recover from an error, then you want at least to have fewest errors. But both of you also said something else implicitly. Both of you sort of had this idea that if the first one did a classification on too few data points, if that would have happened to be a classification where you're very, very, very right, but you do only a tenth of the, 1% of the data, it doesn't bring you very much, right? So I believe, Francesco, I read you to sort of mean that we also have to sort of a balanced split of the data. And there's some truth to that. Ultimately, the question is, how good is the first level? So even if you have a per mil and you don't make a mistake, but still you want to put that on top. So if the difference in terms of performance of this and that is relatively low, then you want to have also a classification that sort of classifies similar amounts. Okay? So it depends a little bit. The more one is better than the other, the more you can put even a classifier in there that classifies a tiny set of all the proteins. The more these four are all having a similar performance, the more what you want on top is something that sort of does half, it sort of splits something in the ballpark of half, third, or something like that, right? Clear? In this particular case, in fact, it turns out that the biology and what we need in terms of machine learning lead to the same situation. So both of those, in fact, are secretary pathway first. So let me sort of introduce some other methods. Again, so there are methods out there that use amino acid composition. There are the signal peptides. P-sort is one that sort of combines signal. And I'm going to go relatively quickly through it. When I go so far, typically for the exam, what is true is the more time I spend on a topic in the lecture, the more likely this will be sampled at the exam. Okay? Now I'm going to go through a bunch of methods. I just want to sort of show that some methods do something else. Does not mean that you need to sort of remember them. Maybe some ideas. And maybe, again, if in the test somebody asks, explain a method that sort of predicts subcellular localization. If you said the name of a method, so what? But what I'd really like you to do is come up with an idea how to do it. Which could actually be describing what some method does. Well, then it's learning by heart again. But it could be your own method. Totally valid. Okay, so P-sort started. Initially, it comes from the Kenta Nakai and Minoru Kanahisha labs. Initially, so it started with the amino acid composition, then brought in all kinds of other signals. So transmembrane segment or not, there's a negative charge or not, there's a cleavage site, there's a signal sequence. So they sort of had a tree of rules, a rule-based system. Put this rule-based system, then came machine learning. There's Paul Horton who sort of introduced machine learning to the idea in P-sort 2 by having a k-nearest neighbor algorithm implied. And then there's a sort of more recent one that uses everything sort of combined what we currently know. And I cannot remember how Paul is pronouncing this. It's not Wolf P-sort. Whatever. But you see where the wolf comes from. And there's a reason in the acronym of the word why it's a wolf. So other methods essentially again use different types of targeting motifs. Here is one that in fact uses Sherloc. The idea is I use motif search, I use phylogenetic profiles, I use some annotation about function, I use some additional data from the genome, I use something that uses amino acid, that uses solvent accessibility, that uses a signal peptide, and we put all of that together into a device that sort of predicts on these different classes. And then there's an extension that does multi-log, essentially it's the same thing, in a slightly different way of sort of splitting the combination of a machine learning. CELLO, the idea here really is in the coding. This is a k segment of length, it's not a k mer. So essentially you're sort of skipping parts in between. So you're looking at residue I and I plus n and I plus m. And essentially look at combined information between those. And it's very tricky to make that work because you immediately see that the combinatorial explosion of the space is such that this can completely easily get out of hand. They allow gaps in between. There's one particular approach that was very interesting. Didn't work very well, but was an interesting idea, I felt. And with this, I get to the point of this slide seems to be wrong. On Thursday, again, there is no lecture. And next week, Tuesday, we're going to go on with the subcellular localization in a different way. What else could we do in terms of predicting subcellular localization? So I gave you a bunch of methods that predict subcellular localization through machine learning devices using different input information. Any idea what else I could put into my input? Those are in the room who have been at other lectures from me last semester. Do you remember? I'm sort of looking into the air, yes.
You input any inputs other than the amino acid fraction? Mm-hmm. Maybe the secondary structure?
That is one idea. I briefly already mentioned that in one of my slides. Yeah, that could be one thing. Yes?
Profiles, we could try them.
So the major issue now here is, in last semester I've always talked about evolutionary information is important. And evolutionary information is important. So one context in which I briefly brought this up in passing for the people who have not been there last semester, in context of the homology-based inference, I showed you the HSSP plot. But what I show here is percentage sequence identity. What I show here is the length of an alignment. And I at some point said, if you have 100 residues aligned between a pair of proteins, and if the percentage sequence identity is more than 33, everything above the line has essentially a similar structure. Okay? Now, the point here is that it means that in a 100 residue protein you can change 67. But when you go to a lab and you change 10 residues in a 100 residue protein, most likely you will change structure. Again, the question is, so this curve says if I'm above that curve, so I change essentially 67, so 100 minus 33 is 67, right? I can change 67 residues and still have the same structure. How come I say now that if I go to the lab and I change 10 residues that I change the structure? How can these two statements be true? Yeah?
We know that what we observe in matrix proteins, that those are the ones that actually work, because all others would have been filtered out by evolution.
Yes.
And if we have random mutations, then those will, with all likelihood, not actually preserve the structure. But those that we see will preserve it, otherwise we wouldn't see them anymore.
Exactly. So what we observe is a tiny exceptional situation. Now let's turn that around. It means that in fact there is only one way to change, there are very, very few ways to change 67 in 100, and there are many, many, many ways not to do it wrong, right? So that means the pattern of what you observe is highly informative for what is relevant for that particular protein. So what can you actually exchange? And that is the evolutionary information. So that's why, in fact, when I look at all of these pairs of things that are above that line, that tells me a lot about the constraints under which this particular protein lives. It's not telling me in the sense that I can put my hand and say this is because the protein is that long or something, but it's something that I can use in machine learning. So far we haven't used that. Now my question is, how can we use it? So far, essentially what we do is we look at an amino acid composition. So we are computing. So the way these methods here have worked. Let me just pull out with one slide where I show the secondary structure prediction with a neural network to illustrate that point. So typically what we do, we have a long protein. From that long protein, we take 13 consecutive residues out, in this particular case. And for these 13 consecutive residues that I take out from a protein, I predict the state, the secondary structure state of the central one. Here's a P. And I ask, is the P in a helix, strand, or other? So it's one flanked on both sides by 6 others, making a total of 13. And then the input here is 13 times 20, because I have 20 amino acids. There's some issue why it's 21. Let's not get into that. But now, profile means you replace that by essentially a count in the family. So in the family, you have my query protein, I have family member 1, family member 2, and so forth. And for the proline here, the set of proline, the query is a P. And then I ask, is this F1 a P, Q, P, or let's have whatever. So I'm essentially asking how many of these residues, in this particular case of the family, have a P here. And I replace this black 100%, and white or yellow 0% by the counts in this profile. Okay, this I can do when I have a cutout of a K-bar, where K is 13 here. How do I do this? When I essentially look at amino acid composition.
I didn't quite understand.
So I believe it's, at least for the three of you, it's clear how we can use profile if we have a sort of K-mer for window-based prediction. But how do I use this same idea of using the profile if I don't have a window like that? What I actually have as input here is 20 units, right? 20 units is the composition of each of the 20 amino acids, for instance, or the surface, or possibly the secondary structure on top. But it's still a composition of everything. How do I put the profile into the story? Yeah?
You could, for example, start by looking at the composition, then looking at the composition of the family, and determine some kind of deviation or surprise level, how the composition changes in comparison to the family.
So this is an unfortunate answer. Let me go a step back. I never heard that answer before, and I never thought about that. It's a great idea. So you may have said that the simplest way in which you can do it is, rather than essentially what I'm doing now is I'm counting the P, and I do that for the entire protein, so the percentage of P in that protein. If I use the evolutionary information, I do the same thing, except I'm not only taking the Q, but I take all the sequences, right? And that would be one way to use evolutionary information. Unfortunately, the reality is that doesn't work. So the simplest generalization of the idea doesn't work. But when we tried this, we didn't have your idea, Tobias. So what Tobias says is, I compute the composition for each one of these each one of these proteins. In this particular case here, 3 for Q, F1, and F2. And then I essentially say, how surprising is the level of similarity of these profiles? Because I assume, again, that F1 and F2, if I threshold it right, have a similar subcellular localization. In fact, I could do homology-based inference. That means, okay, in this particular case, I don't know the localization, but I do know that they are the same. If they are all, remember the point was Q and E, the similarity, the percentage sequence of similarity has to be above a T, a threshold T. Then you can do homology-based inference. Now, you can do that if you choose the right F1s, F2s, and Qs here. then you will know that all three, or all the set that you extract here, will have a similar subcellular localization. And what you essentially expect is then that these vectors, the overall vectors of composition, should be similar because the subcellular localization is similar. That's a cool idea. We should try that. it just never occurred to me. That is a very, very interesting idea. Good. So I don't know whether it would work. Any other idea? Something totally different. How else could I sort of solve the problem? And I'm going to switch off the camera because we're just trying to think outside of the box.